Yeah, Arjun Aditham was already presented, but he's a PhD. He obtained his PhD in bioengineering from Stanford University, working for disease lab, where he developed high-throughput platform for measuring transcription factor DNA binding affinities, and then he moved to a postdoctoral position in Bloom's lab. So we are looking forward to hearing you. So you can start presenting, Arjun. Thank you.
Sounds great. Thank you, Mariano and Adrini, for the introduction. Just before I move on, just want to make sure you just see my slides only and nothing else.
Yeah, I'm seeing it.
Okay, perfect. Thank you. So thank you all for coming today and for the opportunity to speak at the seminar about my in-progress work on doing a deep mutational scan of the rabies glycoprotein. And just as a brief background, rabies virus is a neurotropic virus, so it affects the central nervous system, and it can infect nearly all terrestrial mammals. And in humans, it's almost universally fatal upon symptom onset. Therefore, immediate treatment upon a potential exposure event is crucial. This virus is still a major public health concern and is the subject of several ongoing therapeutic and vaccine development campaigns. And almost all human incidences of rabies virus are unfortunately from dog bites. And I want to emphasize that human exposure to the virus can happen worldwide. And in this map, I'm showing specifically annual human deaths from rabies due to dog bites. So in the Americas, where I'm from, and in Europe, we see almost no deaths. And this is likely due to large-scale mandated rabies vaccinations for dogs and access to therapeutic measures in cases of exposure. However, in other continents, particularly in Africa and Asia, we do see a substantial number of deaths every year occurring. And unfortunately, this virus continues to claim the lives of almost 60,000 people annually, half of whom are children, and deaths are primarily, again, in low resource settings. So upon exposure, a possible exposure to the virus, prophylaxis is, sorry, I'm seeing some stuff show up in the chat. Oh, sorry. Post-exposure prophylaxis for rabies is a two-pronged approach. First, it involves both a local injection of rabies antibodies to stop initial infection of the virus near a bite wound. And additionally, we use a vaccine that requires multiple doses to cultivate our immune systems against the virus. And though there are several efforts to develop new vaccines, today, really, I'm going to be focusing on therapeutic approaches to prevent infections at an exposure or wound site. And currently, we use rabies immunoglobulin. It's typically derived from human serum, and it has a high antibody content to neutralize the virus. And unfortunately, this is prohibitively expensive for many people who need treatment. And to circumvent this, alternative sourcing of serum, for example, from horses, so equine-based treatments have been developed. And although they are substantially less expensive, they have unfortunately carried a higher risk of medical side effects and observed medical contraindications. So because current treatments that use serum pose access issues, there are now efforts to move towards antibodies as an approach for prophylaxis alone. So in 2017, Rabashield, just a single monoclonal antibody, began its clinical use in India. And unfortunately, circulating isolates of this virus can still escape this antibody, and it's limited its clinical use, although it's been able to be applied in the clinic. More recently, given the observed sequence variation that already exists in the virus, many people have been developing monoclonal antibody cocktails. So just as a point of distinction, these cocktails contain 2 to 3 antibodies, as opposed to this initial round of antibody-based treatments, which was just one single antibody. At least 5 of these cocktails are under various stages of development. Some are preclinical and others are in various stages of a clinical trial. So throughout this first portion of the talk, I've basically told you about the rabies virus, its impacts, and how we've tried to therapeutically address it. But now we need to know what part of the virus is being targeted. And All of these therapeutics are targeting generally the viral glycoprotein, which resides on the surface of a viral particle, and it's responsible for mediating viral entry into cells and the broad tissue tropism that is seen with this virus. So, for example, this virus is able to infect muscle cells, and nerve tissues during its early stages of infection and can affect other tissues later on. It's a homotrimeric complex on the viral surface, and I'm just showing that here in various shades of blues to make the protomers more visible. And as I mentioned, variants of this glycoprotein are also observed in natural isolates, complicating the development of therapeutics. So I'm just highlighting in here in red positions throughout the glycoprotein where circulating variants have been observed. And it's pretty clear that they occur throughout the entire protein. So using a technique like deep mutational scanning, where we can measure the effects of all substitutions at all sites of a protein, would really be crucial for developing new therapeutics. And using deep mutational scanning, we can address a ton of questions. But first, we can start by just generating a sequence to function landscape of the glycoprotein, revealing areas that can tolerate mutations and what are possible constraints on the evolvability of this protein. And then we can also study preclinical antibody therapeutics, and namely, we want to identify naturally occurring mutations in the glycoprotein that can escape treatments. And we also want to use our platform to study newer antibodies that have so far evaded direct structural studies, for example. However, deep mutational scanning by itself requires the ability to study thousands of protein mutants in both a time and cost-effective manner. And to study glycoprotein mutants alone, using live rabies virus poses significant challenges to study safely in a laboratory, both in terms of safety as well as just engineering for high throughput assays. So in the Bloom lab, we use a recently developed pseudovirus platform to study the mutational effects of the glycoprotein alone. So for example, we take a glycoprotein that was normally on a virus and we put it onto a barcoded pseudovirus. And this pseudovirus is BSL2, and it's substantially safer. So the pseudovirus genome contains only one viral gene, in this case, the glycoprotein mutant. And for this reason, the pseudovirus is unable to replicate when it infects a cell, like in a laboratory dish. Each genome is also barcoded, so we use PacBio sequencing to link each barcode with its respective glycoprotein mutant. And then in future experiments, we can just sequence the barcodes to link an observed phenotype with an original mutation inside of the protein. And just for purposes of the schematic, I've shown one pseudovirus, but in our assays, we're working with a pseudovirus library. Each particle is barcoded and contains and displays its own unique glycoprotein mutant. And in our deep mutational scan, just as a summary, we studied 8,400 mutants to this protein, and we centered our analysis on the Pasteur strain, which is a lab-adapted strain and is well characterized for our purposes. So then we can start addressing these questions and, for example, just start with generating a sequence to function landscape of this glycoprotein. And to measure mutational effects on the glycoprotein on cellular entry, I did a relatively simple experiment. I took a library of barcoded lentiviral particles or pseudovirus particles. I infected HEC293 T cells and then sequenced the barcodes to quantify each mutant and assign a functional score. So essentially, the more barcodes that are observed in sequencing, the more capable a glycoprotein can mediate cellular entry. And again, these barcodes correspond to a glycoprotein mutant, allowing us to link a functional effect with a particular mutant from our initial library. So to show these results, I'm projecting the average effect of all mutations at every site onto the structure of the glycoprotein. And just for purposes of clarity, I'm only going to show the results on one of the protomers. Sites where mutations are generally tolerated will show up as white on the structure, and those that are mutationally constrained will show up more as orange. And we can see that on several regions of the glycoprotein surface, there are areas where mutations are generally tolerated. However, some regions closer to the base of the glycoprotein are mutationally constrained and cannot handle mutations there. And when we look at the apex of the glycoprotein, so looking from the top down, we can see that there are several more mutations that appear to be constrained and cannot tolerate mutations. And this suggests that targeting therapeutics specifically to the top of the glycoprotein may be an effective strategy to develop therapeutics robust against natural variation. So of course, we're also interested in why certain positions are constrained. So in one case, we can examine which mutations may cause issues with the stability of this protein. So for example, hydrophobic residues are often necessary in internal regions to ensure the folding stability of some of the domains in a particular protein. So we wanted to see in our deep mutational scan if we could infer these effects. So to do this, I just compared the effects of all polar amino acid substitutions relative to hydrophobic substitutions at every single site. And in this structure, regions where hydrophobic residues are heavily preferred are showed in increasing shades of orange. And several of these sites localized to the internal regions of the glycoproteins domain, suggesting that ensuring these hydrophobic interactions are retained in order for the protein to possibly fold properly is a constraint on the tolerance. So we probably could have predicted that just from biochemical intuition alone. So then we wanted to turn our attention to understanding why some unstructured regions of the glycoprotein cannot tolerate mutations. The rabies glycoprotein contains three major domains, each of the fusion domain, central domain, and plexion homology domain, which I've shown here. And they're each separated by unstructured linker regions. And I've only labeled a subset of these that I'll discuss today for purposes of clarity. And on the right, I'm just plotting the histogram of percentage of mutations in a domain or linker, as I'll show, against functional scores. So in the fusion domain, which adopts a structure, we see, for example, that nearly half of the mutations we study cause a loss of function, while almost half have a neutral effect. And for some of these unstructured linkers, for example, there's linkers 2 and 3, which are just on the surface of the glycoprotein, as one might expect, we do see a fair amount of mutational tolerance, and only a small subset of mutants are deleterious to the overall effect of the function of the protein. But then we looked at other linkers, and for example, I'm just going to talk about linker 4 today. Linker 4 is on the top of the glycoprotein, which I'm pointing to with my mouse here. And what we notice is that several mutations at this linker in particular confer several deleterious effects. And in fact, it sort of resembles the behavior to a structured domain when we look back. So to yield insights into why this linker in particular is constrained, we need to look at the other biologically relevant structures that this glycoprotein can adopt. So while I've been showing the pre-fusion conformation of the glycoprotein, during viral fusion into a cell, it must undergo drastic conformational changes in order to function properly. And in this case, linker 4, which I've highlighted up here in teal, undergoes an unstructured to a helical conformation. So currently, we believe that several mutations are deleterious because they directly interfere with this process. And this is a mechanism that others have described prior. And so far, we've seen a couple of ways that these mutations do this. Firstly, it can just inhibit the conformational switch. And by that, I mean, we can introduce mutations that are helix breaking. So it just forces the protein to stay in this pre-fusion conformation because it cannot form a helix for a post-fusion. And then additionally, we found other sites that destabilize interactions that are specifically observed in the post-fusion state that are required. In addition to these helix breaking mutations. So although the linkers appear unstructured, they can also play various critical roles in the overall function of this protein. So in this initial part, I just hope I showed you we can do a deep mutational scan and give you a sort of a flavor of the insights that we're trying to pull out. But now we want to turn our attention towards antibody therapeutics. So in this case, we take our pseudovirus library and we spike in a pseudovirus that contains a glycoprotein from the VSV virus. So unlike the rabies glycoprotein mutants, this VSV is not neutralized by any antibodies of interest in our assays. So we pre-incubate the library with multiple concentrations of antibody. Again, we infect hex cells and we can sequence our barcodes. So in this case, barcodes that persist even at higher concentrations of antibody are signaling an escape mutant from that particular antibody. And again, to emphasize, the VSV barcodes, which I'm showing here in green, are not affected by the antibody. So they should be unimpacted regardless of concentration. So it was a whole process to decide which antibodies to study because there's so many of them. But long story short, we took that whole list and we just cut down to 8 antibodies that were the result of several previously published antibody campaigns against this virus. So 17C7, which is the rabies shield antibody I introduced earlier, is clinically deployed. So we chose to study that one. Several others are preclinical, and others were well characterized, but were either not pursued to begin with, or in some cases were not pursued even or had its clinical trial stopped. So we wanted to study those too. So to just start since it's well characterized, I'm going to just walk through how our escape results look for the 17C7 antibody. And here I'm plotting the average escape score as a function of every position within the glycoprotein's ectodomain. So sites with 0 escape generally do not have mutations that confer escape. And the higher the site escape, the more mutations that generally enable antibody escape. So I took these scores and I projected them onto the structure, and the antibody is in pink, and expectedly the sites with the strongest escape localized to the binding site. So that is reassuring and suggests that our deep mutational scan is recapitulating known information. But we wanted to take a closer look, and just to remind, we can get per mutation resolution at every single site of how it contributes to overall escape. And I'm just showing this here in a heat map where increasing shades of blue indicate higher escape. And for reference, I've listed the wild type amino acid on the top of the heat map. And as I mentioned, the premise of developing antibody therapeutics is that they need to be robust against natural variants. And to look at this on the right, I'm plotting the individual escape score for each mutant. So each of these points is a mutant, and its escape score plotted against its frequency of observation in circulating isolates. So for this antibody, we can appreciate immediately that there are a few mutations here that can both confer escape and are already present in isolates. So for example, positions at arginine 346, it's already well known that isolates escape this antibody. And here, several escape mutations we measured in our DMS are not found in isolates. That's just the nature of running a deep mutational scan that's so comprehensive. But of course, we can see that some of these isolates that are able to escape. Similarly, we found positions at asparagine 336, which can confer escape as well. And interestingly, we found that mutating this isoleucine residue to a threonine can confer a really strong escape effect. And it's worth noting that introducing a threonine may actually cause a glycosylation event to happen at this asparagine 336, and this can suggest that glycosylation of that site may be an escape mechanism. So I just showed you a well-studied and clinically deployed antibody, but in the final few minutes, I just want to provide brief snapshots of two preclinical antibodies, RBC58 and CTB12. So both of these antibodies are part of preclinical cocktails. They also have interesting properties. So in previous course studies, RBC58 and 17C7 were likely to be shown to bind the same site, but we don't have a structure, and we just have modeling of this interaction between the glycoprotein and RBC58. However, RBC58 can neutralize more viral strains than the 17C7 antibody. In the case of CTB12, it's currently part of a phase 3 clinical trial cocktail, and prior attempts to map the escape have failed. And again, these antibodies lack structural characterization, so we wanted to use our deep mutational scan to sort of shed insight into what's going on. So we started with RBC58, and to orient, I'm going to be showing a top-down view of the glycoprotein again, with the escape score mapped onto every site. So progressively bluer positions indicate increased escape at a particular site. So firstly, we can observe that this antibody RBC58 tends to target the top of the glycoprotein. And though I'm not showing this here explicitly, these target sites are highly distinct from the 17C7 antibody, suggesting that despite prior studies, these antibodies actually bind target distinct sites. And again, we can look at escape score versus frequency, and we find that fewer mutations in general escape the RBC58 antibody compared to 17C7, and those mutations that do generally occur at a much lower frequency. So aside from revealing in high detail the escape sites of this antibody, it also sort of correlatively suggests that targeting the apex of the trimer may be an effective therapeutic strategy. And then we wanted to turn our attention to CTB12, another preclinical antibody. And just as a reminder, we don't have a structure of this antibody glycoprotein interaction. And prior efforts to identify the sites from the original paper, that confer escape failed. And again, I'm just going to be showing the results from the top down. And satisfyingly, we were able to use our deep mutational scan to identify escape sites. And again, these escape sites are predominantly on the top of the antibody, but we observed that the antibody targets a unique set of residues compared to the other antibodies we had studied so far. And this residue 270 is part of this linker region that I mentioned earlier in this talks. And again, during viral fusion, this unstructured linker must undergo conformational change into a helix in order to drive viral fusion. So what we hypothesize is that this antibody is partially targeting this linker region, and it's trapping this glycoprotein in its pre-fusion conformation in order to block viral entry. So in conclusion, we use a pseudovirus platform to perform a deep mutational scan of the rabies glycoprotein, and we studied 8,400 mutants that reveal mutationally constrained sites throughout the glycoprotein, and even unstructured regions are mutationally constrained and important for function. We then studied 8 antibodies, many of which are preclinical, to experimentally forecast escape. And importantly, for some antibodies without structural characterization, we're both able to identify target sites and hypothesize their mechanisms of action. But of course, the work is continuing, and this is an ongoing project. And some questions that I'm really interested in is studying, like, why are some antibodies better than others? So as I mentioned, RBC58 is a lot more broad spectrum than 17C7. So we want to look at more examples of that closely. And of course, I love sequence to structure to function questions. So mechanistic characterization is something that I'm really interested in, both for elucidating fundamental principles of how this protein works, as well as applications for vaccine design. So with that, I'd like to thank everyone in the lab, particularly my PI, Jesse Bloom. Kaylin and Caleb were instrumental. Neil King's lab at UW and Naveen were super generous in providing us antibodies. Of course, thanks to our funding sources. And again, I'd like to thank the VESP organizing committee for the opportunity to speak today. So with that, I'd be happy to take any questions.
Thank you, Arjun, for the amazing talk. Very interesting work. One thing that came in my mind is, of course, you are focusing in understanding the mutations of the glycoprotein of the virus. But I think that for entering the human cell, for example, thinking on human cell, you have specific receptors that will bind this glycoprotein, right? Have you thought, or are you thinking of also looking for the mutations in the receptors of the mammal cells, for example, that also could affect the entry of the virus?
So I love that question for several reasons. So rabies utilizes multiple receptors, and there are at least maybe 5 known receptors for this virus, and they play very distinct roles in the infection cycle. So certain receptors are important at the initial infection wound, while others play a role, for example, in retrograde transmission from a peripheral nervous system to the central nervous system. Unfortunately, the sites that these receptors bind on the glycoprotein is not well known. So studying receptors and their mutations, I think, are just important as a technique to map what those interfaces are. We don't have structures for those. But I will say that generally we've observed we've seen that the receptors have a strong sequence conservation throughout several organisms as well. So though we have not explicitly focused on that, I think it's a super valuable question and thing to pursue in the future for just knowing how this thing works.
Okay, great, thank you. We have a question here. Mariano is curious. if you didn't detect any gain of function mutations?
Yeah, we didn't identify any. Most of them are just neutral effects, or were deleterious. Yeah. But
Oh.
again, we're mostly focused on natural isolates here and variation, and we really want to focus on identifying those constrained positions anyways as valuable therapeutic targets.
All right, perfect. And I'm just curious if you were able, like, of doing any cellular experiment to see, like, to manage the antibody escape of those virus. I don't know if you can do this in your lab. So, like, putting an antibody together with a strain that has those most important mutations that you found.
Yeah, so that's we're sort of going about that approach. So what we're really interested in doing now is looking at some of these natural isolates more closely and seeing whether these escape mutations do occur in those natural isolates, as well as seeing whether we can recapitulate that. And I think that would be really powerful to show that our deep mutational scan is actually informative it's actually informative. So while I haven't discussed that here, that is actually another active direction in the lab going right now.
Okay, perfect. And we have one last question here in the Q&A. Are you planning to try mapping immune serum?
That is another great question. Right now, we would love to. There are vaccine trials for the next. So I mentioned that there are new vaccines being developed right now against this virus, and there are trials ongoing. We'd love to get our hands on serum from various sources, of course. And I think it's really one question I'd love to answer is just, does a serum from like an engineered vaccine that is specifically stabilizing the pre-fusion conformation of this glycoprotein different from what we currently use, and what are the antibodies being cultivated? I think that's a great question. Right now we're working with monoclonal antibodies, but I would love to at someday be able to go dig into the serum if we can get any.
Yeah. Okay, great. Thank you. Thank you, guys. I want to thank for our speakers for the very nice presentations of today. And I just want to tell everybody to tune in our next VES on August 6th. Thank you. See you next time. Bye-bye.